It's a pleasure for me to be here and present some of the work we have been done in the Azores. Most of the work that I'm going to present is some research that I've done for my PhD, for doctorado, and which led later to this project, Patelrin, that we developed together with Pablo. And so I'm presenting an overview of all the work that we have been doing with patella. Okay, in the Azores, we have two species of limpets, lapas. We have one which is called patella aspera, and in Portuguese we call it lapa brava. And the other one is patella candii, which we call lapa mansa. They are very easily distinguished by both the shell, and if you turn them around, you can see the color of the foot, which is very different, so they are very easily identified. Also, patella candii usually is a species that occurs intertidally, mostly, whereas patella aspera occurs both intertidally, but also subtidally, so underwater, okay? Limpets are a very important species in the ecosystem because they regulate the amount of algae that grows and occupies space. So they have this radula, which is several rows of teeth, which they use to scrape the rock and remove all the algae that is in the rock. You can see an experiment there. This treatment here where limpets were removed, you can, limpets were not removed, and you can see there is no algal growth. And here, where limpets were removed, you can see there is a lot of fucoid algae. Fucus is a brown, large algae that you also get here in Galicia. So they are important. The problem, in the Azores, this is called, limpets are a delicacy, so it's marisco. So they are the most traditional dish from the Azores in terms of seafood. And there was a very big exploitation, and then the fishery collapsed around 85. There was also a relationship, so islands with more people also tend to have fewer limpets. So there is evidence that there is an effect of over-exploitation, too much fishing on this resource. So this is the start for my PhD. I started from here, and when I know the fishery has collapsed, but how many limpets are there? So I did a survey for both species, and I found that there was regional scale variation. So some islands, for instance, Flores Island has a lot of limpets, Graciosa very few limpets, Pico intermediate levels, San Miguel intermediate levels for candii. Whereas patella aspera, which is the preferred species for people who eat them, is virtually extinct in most of the islands, except in Flores. So there was evidence that there is differences in the abundance of limpets among islands, which is reflected on, which is explained by the difference in population. Okay. The problem, as I told, the limpets play an important role in the ecosystem. So when you relate the cover of algae with the abundance of limpets, you see that there is a negative relationship. So islands with less limpets also have a greater cover of algae. Similarly, they have a lower cover of barnacles. Barnacles are the species that are outcompeted by the large fucoids, okay? So there is a community cascading effect of the exploitation of limpets. This is just to show you some quadrats. This is a quadrat with a very large abundance of limpets, and especially very large ones. This is a 50 by 50 centimeters quadrat, and you can see there is no algal growing there, whereas this is a typical patch of the shore without any limpets, where you can see that algae is already extending and killing all the barnacles. Also, when the algae grow, they occupy space, and if you get fucus, they swipe the rock, so they don't allow the larvae of limpets to attach, to settle. So there is a negative effect. So in this another experiment that I've done, I cleared the turfs, the algae, and the controls where I did not clear the turfs. And where I only got recruitment, settlement of limpets in areas where the algae were removed. So there's a snowball effect. You remove limpets, there are more algae, and then these algae don't allow the limpets to settle. Okay, so there's a snowball effect. Also, I measured a lot of limpets, and you can see that most of the individuals are very small size. You can see this is 1 centimeter, and this was true for the 4 islands that I examined. The dashed line here indicates the minimum size of collection, and as you can see, there are very, very few individuals larger than the legal size of exploitation. So again, another indication that these animals are being overexploited. So we are reducing the abundance and we are reducing the size. Is it the same ecologically? So to test this, I developed an experiment where I had 2 large limpets and 2 small limpets, which I called the normal community. So we have an abundance of large and small individuals, as you would see in a natural shore. And then I exploited this community, but remove only the large ones. So I keep the density, a total of 4 limpets, but they are all small. And another treatment where I removed the abundance, but kept the structure, the size structures. So I still have large and small, but I have a reduction in abundance. And then you can see that the amount grazed is similar between the 2 treatments. So reducing the abundance of animals is having the same effect as losing the large ones. So the large ones are important, okay? We have not tested this, but we know from the literature that a reduction in size also has potentially an impact on the reproductive output. So smaller animals produce less eggs, less juveniles. They contribute less to the reproductive output of the animal, whereas large limpets, large animals, large birds, everything produce more eggs, so they contribute more for the sustainability of the population. Okay, what about marine protected areas? These were shown to be very effective ways to protect, but they also are very important for scientists if you want to see what the community would look like without human intervention, okay? So in the Azores, we also have marine protected areas, and I went to see them. I examined 2 of them and control areas where there is fishing. And what I saw, and I did this work for 3 years, and this is pulling all the data together, so it's the average of the 3 years. And you can see that inside the marine protected areas and outside the marine protected areas, there is no difference between the 2 sites. There's only a difference between time. This is a winter sampling in black, and this is a summer sampling in gray. There is a recruitment during winter, which explains this higher abundance in winter. And then in summer, when the sea conditions are calm, people tend to collect a lot of limpets, and then you can see the decrease in abundance. So there's a seasonal effect, but there's no difference between what's happening within a marine reserve and outside. Okay, so this indicates that reserves are not working. I have the same amount of limpets of animals inside and outside. Why is that? So here I have the average number of people per day per 100 meters of shore seen. And inside, you can see, okay, it's a little bit larger outside the reserve, but you still get some pressure inside the reserve. So there is a lot of illegal collection of animals. So that's why reserves, we believe that this is why reserves are not working. People don't care. So they are reserves on paper, but they are not truly effective reserves because people still go there and collect all the animals. And the Azores is not like the mainland Europe, okay? So the shores are very small space for animals, and one person can have a huge impact on the shore, okay? Here we have very extensive rocky shores, so it's more easy to have, it's more diluted, the effect of people. This is to show that the community, or the picture of the rocky shore in the Azores, and as you can see, there's a lot of algae growing everywhere, even up around the shore, where you have the barnacles, which are these white patches. You can see there is a lot of turfs growing on. This is the typical situation in the Azores that you can find. And fortunately, we have one reserve that is really working, which we sampled last year. And this is it. And you can see that above the low water tide, there's nearly no algae growing. And all these things that you can see are very large animals, very large limpets, okay? The only algae you see is very close to the water, and these holes are made by the sea urchin, Paracenterotus lividus, okay? Ouriço. So, again, here we have a representation of what the community would look like without human intervention. This is very, very important for a scientific, because we have a control, a reference base to know, okay, this is my target. I want to reach this target. Just to say that very large crabs that are not common as well elsewhere are also very abundant inside the reserve. So this reserve is not only protecting limpets, it's protecting the ecosystem as a whole. Okay. Another study that I've done was based on this observation that most of these species, Patella candyi, Lapa mansa, occurs within cracks, pits, holes. So, and I say, okay, let's see if this is really, why is this? So I sampled and I saw that there was a significant association of the animals with these features of the rock. So there were more animals inside holes and pits and cracks than expected by chance, according to the abundance of the crevices and the open rock. I also found, I put some tiny tags on limpets that were inside the holes and those that were outside. And I followed them through time, and I saw that the ones that were outside the holes died more than the ones that were inside the pits. So again, this was showing that the holes, the pits, they confer the species some protection from predation, desiccation, and all, or even exploitation, because it's more difficult to collect a limpet which is inside a hole. I also found that there was differences in the size of the animals that were inside the holes and the ones that were on open rock. And the ones that were inside the holes were smaller, okay? This indicates that these microhabitats may be more important for juvenile stages of the animals, not so much for the adults. Okay. This is very interesting, but is this information of any relevance? Is there any conservation value for this information? What we know is that human populations tend to be concentrated on the shore, on the shoreline. And as a consequence of that, we are replacing thousands of kilometers of shoreline by urban structures. You probably have them here as well. So, seawalls made of concrete, pontoons, everything, wharfs, everything. We are replacing the natural habitat by artificial habitat. And what happens is that if you compare both, this is a seawall, and you see it's very, very flat, very homogeneous, whereas a natural rocky shore is lots of microhabitats. This is at large scale. If you look at small scale, this is concrete. It's very smooth, no cracks, no pits, no holes. A natural shore usually have a lot of these features, okay? So in the Azores, we also have these kind of seawalls which are made of basalt, but the rock is quarried inside the island and is very flat. So you can see a picture here, there's only a few barnacles, which are these white dots here, and then there's a black rock, nothing else there. Okay, so this is a problem. What if I drill, if I do some holes in the seawall? What happens? That's what I've done. And I've drilled large holes, small holes, very abundant, very numerous holes, few holes. And what I found was that generally there were more animals in areas where the habitat has been enhanced, where I drilled the holes, than in control areas, okay? So it's showing that by drilling holes, increasing the topographic complexity of the seawall, of artificial substrate, I'm increasing the abundance of limpets, okay? And I also found that there were differences between the treatments. So for the recently recruited animals, it was more important the number of holes that are drilled. More holes, more animals. For the juveniles, the results were inconclusive. And for adults, what was important was the size. They were responding to the size. Of course, a large limpet cannot fit in a small hole, so they were limited by the number of large holes. So these results suggest that habitat enhancement was successful at increasing the number of limpets on these man-made structures. So we can modify them very easily to use them for conservation purpose, for instance, okay? So also very important, there was evidence that I increased the settlement, the recruitment, which is very important if you're considering the management of a fishery. You need recruitment. This is the important. This is the replacement of the population. So you need measures that increase the recruitment. And this is just to say that this is a very simple thing to do, which can be used and conciliate our need to protect the shorelines and conservation, okay? This is just to show you some pictures where you can see clearly that the limpets are inside the holes, the large holes, the small holes, everywhere. So clear they really like these things. Okay, so there's a big fishery about limpets, but we don't know if they are any good for health. Can we eat them safely? We don't know. Also, volcanoes, the Azores are volcanic islands, and still the volcanoes are still active. And volcanism is a source of natural pollution, okay? It releases a lot of things that are nasty, okay? Is this having an impact on the things that we eat? It's important to know. So we sampled 2 areas. This is San Miguel Island, and we sampled 2 areas that have shallow water vents. These are areas where there is natural emission of gases, of volcanic gases. So if you go there, you see bubbling from the sea, and the water is warm, okay? And we have 2 of these, Ferraria and Ribeira Quente, and then 2 control locations where there's no emissions of volcanic activity. Okay, and what we found was that there were some differences between the areas with volcanic activity and the ones without volcanic activity. One of them was the amount of calcium found on the animals, which is very easy to interpret because the emissions of the volcanic emissions reduce the pH of the seawater. In enclosed bays where there is activity, you can get a pH of 6, which is very acidic compared to the normal seawater, which is around 8. So, and the pH destroys the calcium, okay? We also found differences in the amounts of manganese, I don't know how to say in English, and stroncio. I suspect Spanish should be similar. So there's an effect of volcanic activity, but none of these are not limiting. What we are concerned about for people is the amount of mercury, lead, and cadmium. And for mercury, it was okay, lead was okay, but for cadmium, we saw that it changed from place to place, but you can see that there are some places where the amount of cadmium is nearly 3 times more than the one that is established by European Communion levels, okay? So we have more concentration of some heavy metals on animals than that one that is legal by European laws. And cadmium can have some negative effects on human health. We also found that animals that were collected in areas with volcanic activity usually showed a much more nuclei that were dead, so which is called apoptosis, okay? So there's an indication that they live in a stressful environment. This is not good for them. And we also found that in Patelocaniae, which is a gonochoric species, which means that it has males and females separately, is not hermaphroditic. We found more hermaphroditic animals in these areas, which is an indication that the volcanic activities are messing up with the hormones and things like that, okay? Okay, epibiosis is the study, or is not the study, many species use other species as a habitat, and this is called epibiosis. And usually this is a clean shell of a limpet, but usually you never find it like this for Patelaspora. Usually it's heavily colonized by other organisms, mostly algae. And what we have done with all the animals that we collected for the project that Pablo is going to talk, we examined the shells, okay? So, and we examined 700 shells, and we found an astonishingly high number of species on the shells, okay? So you found nearly 200 species of algae and animals living on the shells of the limpets. Of these, 17 were new species never recorded in the Azores. They are not new species for science, but they are new species for the Azores, okay? So we also found that there was a very highly significant relationship between the size of the shell and the number of species that it supports. So the greater the shell, the larger the shell, the more species it can support. So overall, these results suggest that limpets are also an important habitat for other species, the shells, and that especially the larger animals, okay? We are also interested in the reproduction of these animals, and there's a PhD going on. This is preliminary results, and where we are comparing the reproductive stages all throughout the year now, and we are comparing that to data that we have from the 80s, so nearly 30 years ago. And this is important to see if there is an effects of climate change and stuff like that. And what the preliminary results are showing is that nowadays data, there is a lack of individuals reaching the highest level of maturation, okay? We don't have as many big and fat animals, whereas 30 years ago, there was a clear peak in reproduction in winter, okay? So this suggests that there's something going on, that less animals are reproducing, okay? We still need to confirm this result because this is only 1 year. It could have been an abnormal year. So we are repeating these 2 more years to see if there is a consistency in these results. But if this is true, this is showing that there is a lack of reproduction going on, which could affect the sustainability of the populations. So all these work suggests that there is a problem with limpets, and there's a problem in marine reserves. They are not effective because people still go there. But we have a lot of marine reserves, and only a few people to go there and supervise if they are okay. So maybe if we reduce the number of marine reserves, we have better enforcement. It's better. I'd rather have 2 reserves that work than 20 reserves that don't work, okay? Because I need them to work. So in order to do that, we need to know if there is connectivity among populations. And that's where we designed an experiment and submitted a project which is called Patelgene, which is just using the genetic data to help us to identify if there is larval connectivity and at what scales, what distances should I put my reserves, okay? Can I have 1 reserve for the entire archipelago? Or do I need 1 reserve per island? Do I need more reserves per island? That's the kind of questions that we are concerned.
Después de la interesante presentación de Gustavo, que aborda la parte ecológica y reproductiva, voy a contaros lo que hicimos desde el punto de vista genético. Bueno, desde un punto de vista conservacionista, lo primero que tenemos que definir es qué cosa hay que conservar. Hay que acotar la unidad biológica. De lo que partimos es de esta estructura construida con caracteres morfológicos, es decir, la taxonomía clásica, que reconoce en el género Patella 9 especies. Estas 9 especies solamente existen en el Mediterráneo y el Atlántico. Son especies que se han formado por especiación y diversificación en el Atlántico después de la apertura del mar de Tetis, la ruptura de Pangea hace 21 millones de años. Solamente existen en el Atlántico y Mediterráneo, ¿de acuerdo? Hay otros géneros de la familia Patellidae, pero no están distribuidos en nuestras costas. Entonces, lo que podemos preguntarnos es si la morfología nos está contando toda la verdad sobre la estructura génica de estas especies, si estos genomas son íntegros o están subdivididos, si tenemos que conservar todo o parte de eso. Y como ejemplo de la incertidumbre que existe, os pongo esta diapositiva de Patella candei, una de las especies distribuidas en Azores, que Weber y Hawkins en el año 2002 identifican con todos estos morfotipos. En principio dicen es la misma especie, pero es una especie que adopta todas estas formas. ¿Vosotros diríais que es la misma especie o que no es la misma especie? En principio, morfológicamente parece que no, ¿verdad? Bueno, pues ellos describen esto, proponen que puede haber subespecies, pero esto no se puede responder hasta que no se aplican marcadores genéticos que nos cuenten el estatus real de estas especies. Como ejemplo de la incertidumbre que causa la morfología, tenemos estas 2 formas, mosca y mansa, ¿de acuerdo?, distribuidas en San Miguel y Santa María, en el archipiélago de las Azores. Un estudiante hace años, dirigido por un grupo de investigación de allí, se planteó saber si esas 2 formas eran especies distintas o no. Una es de la zona intermareal más emergente y la otra es de la zona, ¿cómo se dice, Gustavo? La más inmersa, la submareal, perdón. ¿De acuerdo? Al aplicar marcadores alocímicos, concluye que mosca y mansa son formas de Patella candei gomesii y que probablemente representan la misma unidad reproductiva, es decir, es la misma especie desde el punto de vista genético y por tanto todas las variaciones morfológicas que estamos viendo son debidas al efecto del ambiente sobre un genoma plástico, ¿de acuerdo? Bueno, pues entonces, si eso es así, tal vez aquí haya una sola especie y haya variaciones morfológicas adaptativas de un mismo genoma a distintos ambientes. Cuando se aplican marcadores genéticos a estas formas, ya en los años finales de los 90, se descubren cosas nuevas. Por ejemplo, se descubre que, por ejemplo, lo que era Ulysipponensis, que era patela distribuida en el continente, ahora aparece una nueva forma que es patela áspera, típica de las islas macaronésicas, y lo que era patela candei, una especie también distribuida en las macaronésicas, que corresponde a la fotografía anterior, se podría subdividir en 4 subespecies: candei gomesi, ordinaria candei y crenata. Bueno, esas formas o subespecies se distribuyen gomesi en Azores, ordinaria en Madeira, candei en Selvagens y crenata en las Islas Canarias. Cuando aplican los marcadores alocímicos, que son marcadores proteicos pero de determinación genética, combinados con la morfología, concluyen que candei de las Azores difiere de candei de Madeira y de Canarias, con lo cual aquellas formas que veíamos distintas también tienen un reflejo en el genoma. Genéticamente, con alocimas también son diferentes, ¿de acuerdo? Estudios posteriores, aumentando el número de marcadores genéticos, confirman esa existencia de distintas formas en distintas islas macaronésicas. También llegan a la conclusión de que candei es la forma ancestral, gomesi es una forma relicta que habita solamente en Selvagens, y destacan 2 grupos principales: gomesi candei, que son estos 2 del punteado más oscuro, y ordinaria y crenata, formada por las que habitan Madeira y Canarias. Por tanto, lo que podemos decir es que en cada grupo, en cada archipiélago, habita una forma distinta de un genoma común, que es el del patela candei, que podría tener el estatus de subespecie. Cara a la conservación genética, tenemos claro que si son subespecies distintas, son genomas distintos y que habría que conservar esos genomas, independientemente los unos de los otros. Cualquier pérdida implicaría pérdida de diversidad adaptativa, ¿vale? Pero a efectos prácticos, cada una de estos archipiélagos no están gestionados por las mismas administraciones y en ocasiones no lo son por los mismos países, ¿de acuerdo? Portugal gestiona las islas que le pertenecen como país, Canarias pues el Estado español, e incluso dentro de cada uno de los archipiélagos hay un propio gobierno que dicta las normas y financia proyectos y dicta las regulaciones de pesca. Por tanto, tenemos que descender al nivel de conservación genética de las formas específicas que existen dentro de cada uno de esos archipiélagos. ¿De acuerdo? Como comprenderéis, la complejidad de la gestión para la conservación ya no va a depender solamente del estatus biológico, sino que va a depender de los intereses políticos y económicos de cada uno de los archipiélagos, con lo cual se complica diseñar una política de gestión común para todos ellos. Bueno, en trabajos previos al nuestro, con marcadores genéticos ya apuntando a los archipiélagos concretos, a las unidades de manejo administrativo, se encontró que, bueno, con una ANOVA, un análisis de varianza, pues nos dice cuánta diversidad genética hay entre grupos y cuánta, por ejemplo, entre el grupo 1 de Azores y Canarias tenemos una diversidad genética muy alta, un 5%, mucho mayor que entre poblaciones dentro del grupo. Bueno, eso nos está diciendo que entre esos 2 grupos sí que hay una diferencia potente, que era lo que esperábamos, puesto que hay formas o subespecies diferentes. Sin embargo, dentro del grupo del archipiélago de las Azores, las islas del norte, Flores, las centrales y las del sur, la divergencia entre grupos es muy pequeña, lo cual hacía apuntar a que no hay realmente una diferenciación genética dentro del pool de las Azores, que se podría considerar todo él como un solo pool génico susceptible de ser manejado en programas de conservación como una unidad de manejo, ¿vale? En Patelgin nos planteamos los siguientes objetivos. Utilizar muestras que sean representativas de todo el pool génico del archipiélago de las Azores, puesto que lo que se había utilizado anteriormente eran muestras reducidas. Entonces no puedes concluir cosas contundentes con muestras pequeñas. También nos planteamos utilizar marcadores microsatélites, que sabéis que son los más resolutivos dentro de especie, para estudiar un genoma y ver cómo se distribuye la diversidad de ese genoma en el espacio. Pero además que fuesen específicos de especie. Anteriormente se habían utilizado, pero tomados prestados de otras especies, con lo cual tienen algunos problemas técnicos y algunos artefactos. El tercer objetivo era elaborar un patrón de conectividad robusto. Con robusto quiero decir establecer la direccionalidad de la conexión larvaria entre las islas, la periodicidad, si se produce en determinadas estaciones del año, y la intensidad, cuántas larvas pasan de una isla a otra. ¿De acuerdo? Si tenemos establecido ese patrón, podemos diseñar un plan de manejo en función de esos datos ecogenéticos. ¿De acuerdo? Plan de manejo ajustado y adaptado al ciclo vital de esas especies. Bueno, pues el primer punto era el muestreo. El muestreo que efectuaron los componentes. Aquí está yo ahí muestreando lapas. El muestreo es importante. En cada una de las 9 islas se muestrearon 2 especies, Patella candei y Patella áspera, que son las únicas que se distribuyen en este archipiélago. Cada color responde a una de las 2 especies y esos 105 pues son 2 muestras de 51 réplica, ¿vale? Con lo cual esto se repite en todas las islas y el muestreo entonces es importante, casi 1000 individuos de cada una de las especies. Con este muestreo pues vamos a ser capaces de concluir cosas importantes. El segundo objetivo era el estudio de microsatélites como marcadores hipervariables del genoma, pero no existían y por tanto a partir del proyecto PatelGene aislamos marcadores microsatélites utilizando, bueno, pues como sabéis, las nuevas técnicas de secuenciación masiva que te permiten aislar miles de marcadores pues en 2 o 3 días, ¿no? Se hacen las genotecas para cada una de las especies. Bueno, no os cuento detalles técnicos y al final obtenemos pues unos 300 marcadores para áspera y unos 100 marcadores para candei. Como punto de referencia, la gente está utilizando entre 6 y 10 marcadores. Se estima que estadísticamente hay que utilizar al menos 15 marcadores, entre 10 y 20, para que no haya problemas estadísticos en cuanto a los patrones de conectividad que estableces, ¿vale? Por tanto, nosotros tenemos de sobra marcadores para abordar estos estudios. Bueno, esos marcadores se multiplexan con distintos colores, cada uno, de manera que en la PCR podemos amplificarlos simultáneamente y podemos tener datos genotípicos, heterocigotos, homocigotos, heterocigotos, ¿vale? De muchos marcadores en una misma migración electroforética por capilaridad. Bueno, tenemos los marcadores y vamos a aplicarlos a Patella áspera. Esta es una representación de el número de pools génicos o de unidades génicas o de unidades de manejo desde el punto de vista genético que existiría en las 9 islas, desde Corvo hasta Santa María, en la especie Patella áspera, haciendo una reconstrucción bayesiana con a prioris y con los datos genéticos. Como veis, el color implica homogeneidad genética en todo el archipiélago. En Canarias, que tendríamos la Patella candei crenata, veis como la composición del genoma ya tiene un color diferente, además del genoma de áspera de las Azores, y por tanto va a ser otra subespecie, y ya la especie del continente de Lisboa es Patella ulisiponensis, que se creía que era la misma que áspera, ¿de acuerdo? Bueno, pues estos análisis nos permiten decir, bueno, pues en principio parece que hay un solo pool génico en todas las islas de Azores. Cuando vamos a Candei, a la otra especie que se distribuye en las Azores, tenemos un escenario muy similar. En rojo es el genoma de Candei. En verde ya tenemos Canarias, que ya sabemos que es otra subespecie. Entonces, en principio, la integridad genética de estas 2 especies en el archipiélago de las Azores es bastante elevada. Desde un punto de vista conservacionista es una situación muy favorable, puesto que si tenemos una homogeneidad genética y no hay distintas unidades que conservar, con un solo plan de gestión serviría para proteger la mayoría de la diversidad de la especie. Podemos concluir entonces que las poblaciones de áspera en las Azores constituyen una metapoblación con movimiento migratorio de larvas entre las islas que homogeniza el pool génico. Pero además, a partir de esos datos genéticos que están ahora bajo estudio, también vemos que el tamaño efectivo poblacional, que es el tamaño que explica la variabilidad genética, el número de reproductores que explica esa variabilidad, es muy pequeño, está muy reducido. Como es de esperar tras décadas de sobreexplotación, y también la sobreexplotación lleva a que haya pocos individuos, hay un efecto de deriva génica y entonces los descendientes ya son parientes entre sí, con lo cual se crea una endogamia que aumenta más, o sea, que disminuye más el tamaño efectivo poblacional. Bueno, las cuestiones que estamos intentando resolver ahora son estas. ¿Hay escalas espaciales en las que el flujo génico sea distinto entre islas? ¿Hay alguna isla que tenga menos flujo génico con las otras? ¿Hay zonas de alta diversidad genética dentro del archipiélago que podamos decir, oye, pues ahora estas islas de esta parte van a ser los reservorios genéticos de la especie y vamos a protegerlas mientras que vamos a permitir cierto grado de explotación en las otras? ¿Cómo es el patrón de migración entre las distintas islas? ¿Es unidireccional? ¿Es multidireccional? Bueno, la direccionalidad nos lo tiene que decir tanto los marcadores microsatélites, puesto que con programas bayesianos podemos establecer la direccionalidad de la migración, como los estudios de dispersión larvaria que utilizan los ecólogos. La periodicidad, pues simplemente con réplica de los experimentos entre distintas estaciones, y la intensidad también nos la dan los marcadores genéticos. Parece, según los últimos datos, que el flujo génico en esta zona de las islas centrales es muy elevado, un poco más atenuado con las islas del sur y más atenuado aún con las islas de Flores, pero resulta que las islas de Flores, como os dijo Gustavo, también tiene una mayor población de áspera, ¿verdad? Con lo cual es la candidata a ser el reservorio natural génico de esta especie en todo el archipiélago. Si conseguimos un plan de protección de las patelas de Flores y la conectividad se produce de forma natural hacia el resto del archipiélago, siempre podemos asegurar la persistencia de ese flujo génico y de las especies, de las dos especies de lapas en todo el archipiélago. Bueno, el último objetivo, y ya termino, sería elaborar un plan de manejo fundamentado en estos datos ecogenéticos, que sea efectivo y que permita pues, que este maravilloso enclave natural de Europa, uno de los más extremos, pues siga manteniendo esas poblaciones endémicas de candei que solo existen ahí y que se siga manteniendo el equilibrio trófico, que se impida esa proliferación brutal de algas, que se mantenga la biodiversidad que habita sobre las conchas de las lapas, etcétera, etcétera. Pero esto va a tener que ser ya en una próxima charla, porque el proyecto termina la semana próxima. Y como sabéis, no siempre que termina el proyecto tienes todos los datos elaborados ya, ¿no? Estamos ahora con el procesamiento de los datos y bueno, pues interpretando y viendo, poniendo sobre todo en conjunto los datos genéticos y los datos ecológicos para hacer una interpretación común. Y nada más, muchas gracias por vuestra atención. Si queréis ver los resultados del proyecto que iremos subiendo, y hay cosas subidas, tenéis ahí en patelgym.com todos los datos. Muchas gracias.